Thank you so much for the introduction and for the invitation today to come and tell you about my work. So as Sonia said, I'm Emily Gehrels. I'm a new CNRS researcher and a group leader at the Interdisciplinary Nanoscience Center in Marseille, France. And today I'll be talking about the work that I did during my postdoc in the group of Thomas Lecrie at the Marseille Developmental Biology Institute. So I'll tell you today about tissue flow, which is required for the creation of biological form. And specifically, I'll tell you about a symmetry breaking event that takes place in the early Drosophila embryo, which creates directional tissue flow. So tissue flow is a critical part of embryonic development, regardless of the species. So for example, here we're looking at a chick, Xenopus, and Drosophila embryo during embryonic development. And what we see in all of these cases is that cells need to be able to move to allow the tissue to reorganize and fold to create the structure required to form the adult. So my research is motivated by a desire to understand the physical mechanisms that drive these tissue flows. So in order to understand the dynamics of the tissue, we need to consider the physical properties of the cells that make up the tissue. So here I have a schematic of a tissue that's just composed of cells that are packed together. And the cells can have differential properties. So cells can have elasticity and viscosity that are responses in response to external stresses. There can be adhesion between different cells and different neighboring cells stick to each other or not, depending on the cell type. And different cells can have different levels of cortical tension. So this is just a line tension that acts kind of as a surface tension along the edges of each of the cells and drives them to want to compact and stay tight. So now if we think about what could generate tissue dynamics or movements of tissues in response to these different properties, you can imagine that if you have a specific region of the tissue where the cells have higher cortical tension than their neighbors, these cells will want to contract more than their neighbors. They'll pull more strongly. And depending on the elasticity and the viscosity of the surrounding cells, you could have the surrounding cells deform in response to the pull, or you can have them realize a viscous flow that actually propagates to much further neighboring cells and leads to long-range flows in response to this increase locally in cortical tension. So we really want to understand how local changes in cell properties can impact these tissue flows. And to do that, we, as I said, work in the Drosophila. So I'll introduce you to the Drosophila embryo. So here's an embryo. We see that the embryos consists of a single layer of cells that surround a fluid yolk interior. So what you're seeing labeled here fluorescently are the cell membranes. So we're seeing a cut through of each of these cells that are columnar. So they're long in this direction and hexagonal in the other axis. And surrounding this entire embryo and giving it its shape, but you can't really see it in this image here, there's a rigid eggshell that holds the embryo in this elliptic shape. So at the beginning, the embryo starts out looking pretty uniform and without much structure. But if I let this video play, you'll see that right at this time, the cells suddenly start to flow and move and fold and create the structure that I was talking about earlier that's required to do the whole process of morphogenesis, to initiate it. And notably, the very first movement is this patch of cells on the posterior or left-hand side of the embryo flows upwards and to the right and begins to bend inwards. So we're interested in understanding really this first phase of flow of the tissue. So in order to understand what's driving it, we first want to quantify the flow. So what we see is at early times, this flow starts out symmetric. So there's a clockwise flow of tissue in this left-hand side of the embryo. And there's a mirror image of that flow on the right-hand side that creates counterclockwise flow. And so we have this symmetric phase of flow towards this top region of the embryo. But just a few minutes later, the profile of the flow changes dramatically. And then we have almost uniformly throughout the embryo, clockwise flow. So we switch from a symmetric phase of flow to a polarized phase of flow. And we can quantify this transition by taking the average of the velocity around the embryo. So if we consider clockwise arrows as positive and counterclockwise arrows as negative, and we average around the closed loop of the embryo, we can see that at early times we have zero average flow or symmetric flow, and then we see a sudden transition to polarized flow that gets more and more polarized over time. So we're really interested in understanding what forces drive the flow in general, this first phase of flow that's really important for morphogenesis, and specifically what drives this transition from symmetric to polarized flow. So if we think generally about forces in biological systems, maybe most of you are used to thinking about myosin. So myosin is a motor protein that can walk along actin filaments. And if you have a dense network of actin filaments within a cell where the actin is kind of binding to the edges of the cell, and then you add a bunch of myosin to this network, you actually end up in a situation where the myosin walking along the actin filaments causes the whole network to contract. And this ends up actually having an effect of increasing the cortical tension of the cells that I was talking about earlier. So again, you have cells that have more myosin than their neighbors, they want to contract, and that can lead to long-range flows. So we want to look at our embryos and see where the myosin is and if it can correspond to the flows that we're seeing. So we take the same types of images of our embryo that I showed before, but now instead of looking at the cell membranes fluorescently, we look at the spatial distribution of myosin. And what we see at early times is that there's a lot of myosin on the top half of the embryo and much less on the bottom half of the embryo. And this kind of intuitively matches what we would have expected, because if you consider myosin again as just contractile, this region of the embryo would be contracting and dragging all of the neighboring cells towards its region of contraction. So intuitively, this makes sense, but we can also write down a simple 1D continuum model where we take into account the active tension of the different cells that varies in space. So this active tension is just that cortical tension I was talking about earlier. There's some spatially varying, there's some viscosity of the tissue, and there's also some friction that is just any interaction between the tissue and the surrounding. And I should say that this model and all of the modeling and simulation that I'll show in this talk was done by Badman Chakraborty and Matthias Merkel, my collaborators for this project. So we can now take this very simple model and use the experimentally measured myosin profile that we see and fit the velocity profile to the experimental data that we see. And we see that we get a pretty good fit. And specifically, we see nicely this region of clockwise flow in the posterior and counterclockwise flow in the anterior on the left and right half of the embryos. So this is nice. Can we test experimentally by changing the myosin profile and changing the symmetric phase of flow? We can do that. Again, I told you that the important feature that drives this flow is the difference in myosin on this dorsal and ventral side of the embryo. So we can use a mutant that gets rid of this difference in dorsal and ventral myosin. So in fact, it totally gets rid of the coordinate system in the embryo that tells the embryo that it has a top and a bottom. And in this case, you see that the myosin is the same on the top and the bottom of the embryo. And if we look at the flow arrows here in white, we see that we've totally suppressed this early phase of symmetric flow. So that's great. We understand that myosin could be contractile, it can drive symmetric flow. But as I had mentioned earlier, what we're really interested in is understanding this transition from symmetric to polarized flow. So now if we look at the myosin profile at this later time, when we have the onset of polarized flow, we see that globally the profile of myosin doesn't look that different. And the main difference is that we have this new patch of myosin on the posterior of the embryo here that appears just around the same time that the flow transitions from symmetric to polarized flow. So the obvious hypothesis that we had was somehow this appearance of this new patch of myosin is causing the transition to polarized flow. And indeed, we can do a mutation where we get rid of this patch of myosin on the posterior of the embryo. And by averaging velocity around the outside of the embryo, we see that we totally suppress this polarized phase of flow. So it is this patch of myosin that's driving polarized flow. But now if we go back to our model and we add in a new patch of myosin in a different location, we see that this model predicts that we should have still a symmetric phase of flow, but just centered around a different region of the embryo. And indeed, we can try to fit our experimental data, our model to our experimental data, and we see that it does a terrible job. And that's because this model, if you average it around a closed loop, will always give you zero average flow. There's no term in this model that could give you a flow that has some polarity. So then we decided to go in experimentally and try to knock out a bunch of different features of the embryo to see what we could do to remove this polarized flow. So the first thing that came to mind is that we know that there's cell scale polarity in the embryo. So we know that there's some initial genetic patterning of the embryo that allows the cells to actually know where they are in the embryo and to be able notably to tell left from right and up from down. And so we thought, maybe these cells are actively crawling or moving in one direction specifically. Somehow they're sensing the direction that they need to go and they're actively moving that direction. And we know that this feature of cell scale polarity can definitely play an effect on later phases, even just after this onset of polarized flow. So we thought it was a logical idea that somehow this cell scale polarity was playing a role. But when we totally suppress cell scale polarity and a couple of other things also, the polarized flow still happens and happens pretty normally, at least for the first, 20 minutes of flow that I'm showing in this video. So then we said, okay, so the cells themselves maybe aren't crawling in a specific direction, but we know that there's a region in the embryo where there's actually adhesion between the cells of the tissue and the eggshell that's surrounding it. So it's in this region. We know that it occurs a little bit later than this, but not much later in the embryo. So we thought, maybe because the cells are sticking here and there's a patch of myosin here, the whole region of the tissue is getting pulled towards this region of adhesion, or maybe somehow the cells here are grabbing on and crawling along the eggshell and pulling the tissue with them. So again, we got rid of this adhesion between the tissue and the eggshell. And again, the polarized flow, at least for these first 20 minutes, was really hard to see any difference between that and the control. So, okay, it's not cell scale polarity, it's not adhesion. And we know that it's related to this patch of myosin on the end of the embryo. So we thought, what if we just make this patch of myosin on the end of the embryo bigger? Then maybe we can accelerate the flow because we have more of this contractile force-generating material. So let's expand the posterior domain of the embryo. Let's put more myosin in. Let's get more polarized flow. And what actually ended up happening is, as you can see here, there's still polarized flow, but it actually turns out that the polarized flow is delayed and slowed down, even when we have more myosin. So clearly, there's something that's missing from this very simple model. And that's not necessarily totally intuitive about how this myosin is generating this polarized flow. And that's not very surprising. This is a simple 1D continuum model. We're taking into account, only these three different factors. So we started thinking, what do we need to add into the model that really might be contributing to the physical behavior that we're seeing? So the first thing we thought about is that the continuum model is 1D. And so it's kind of assuming a flat line with periodic boundary conditions, but that's really not physically matching with what's happening in the embryos. And specifically, as I told you, there's this rigid eggshell that's constraining the embryo and holding it rigidly in the shape that it has, this elliptical shape. And so it's maybe important to take into account the varying geometry of the embryo, the fact that it's highly curved at the two ends, and that in the middle it's actually quite flat. And the other thing that you can see is missing from the model when you look at the images is, again, the model assumes that it's a 1D line that's now closed into an ellipse, but the cells actually, aren't a flat line. They have some height. And if you look at the myosin and where the myosin actually is within the cells, I told you that myosin is acting to just want to contract the cells. but that assumes that myosin is uniformly expressed throughout the height of the cells. And when we actually measure the myosin, we see that there's almost none within the cytoplasm of the cells. It's really localized only on the basal side here in the inside of the cells here, and on the apical or outside of the cells here. And now if we think about what happens if you have a force-generating molecule that's specifically localized to one side of the cell versus the other, what that does is it creates a torque in the tissue as the top of the cells want to contract and the bottoms don't want to contract. And so the tissue actually ends up bending inwards and experiencing this torque. And indeed, if you look at the images of the embryo at the time when you have this patch of apical myosin in the end, you see that the tissue is bending inward. So we know that this torque is actually acting and is present and important in the system. So what happens if you take into account these two different features of the embryo that we've up to this point neglected? So first, we can think kind of intuitively about what happens if you have a patch of cells that has myosin on one side and wants to bend inward, and there's an embryo that has varying curvature around the outside. So first, we can put this patch of cells on the region that's highly curved. So these cells want to bend inwards, but they actually have to overcome the original outward bending of the cell of the embryo in order to reach their kind of preferred inwardly bent state, as compared to cells that are in this flat region of the embryo that have a much easier time bending inwards and don't have to overcome this initial kind of barrier. So intuitively, you might think that if we're giving emotions to the cells, that they would want to move to this region where it's easier for them to deform in the way they want. That's kind of some intuition that says maybe these cells want to flow that way. But now what we can do instead of just using intuition is take our original model and rederive an additional term that takes into account this spatially varying curvature of the embryo and this active moment that is related to this torque that I was telling, and it is just related to the difference in myosin on the top and bottom of the cells multiplied by the height of the cell. So now we can take this updated model with this new term, we can fit it to our data, and now we see, great, we can predict polarized flow. So this equation is now no longer bound to be to give only zero average velocity. We can have regions that are clockwise that aren't balanced by regions of counterclockwise flow. So it's very nice. We have a model that seems to match our just normal control data, and we can actually take the ideas of this model. Again, my collaborator Banan Chakraborty created a vertex model simulation where he takes an elliptic embryo surrounded by a rigid eggshell and gives it some patch of myosin. And he sees that just allowing the embryo to equilibrate to its preferred shape leads to this type of clockwise flow that we see experimentally. And notably, if you get rid of the gradient curvature, you have a perfectly round embryo. The patch of myosin won't move because there's no gradient and curvature for it to flow down. So we wanted to test this experimentally. So we did that using these embryos that are called FAT2. So the embryos are much roundier than wild-type embryos. They're not perfectly circular or spherical. We would love that, but the mother flies actually can't lay perfectly spherical embryos. They get blocked as they're trying to lay them. So this is the best we can do, but it's still great. We have a much decreased gradient and curvature. And what's nice about these embryos is that they still do exhibit pretty normal flow. And actually, just as a side note, the embryos themselves are not mutant. So these are kind of wild-type embryos. The only thing that's mutant is the geometry, and we do that by doing a mutation within the mother in the cells that create the shape of the embryo. So all of the myosin distribution should be pretty normal in these fat embryos. So now we can measure the flow in these embryos, and what we see is compared to wild type, they still have a polarized flow. You can see it in the movie, but the flow is slowed down compared to the wild type flies. And now we can do simulations where we take embryos of varying aspect ratio, where this 2.5 aspect ratio matches pretty well with the wild type value. The aspect ratio of 1 would be a perfectly round embryo that has no polarized flow. And for some intermediate value, you see this decreased onset of polarized flow as we see in our experiments. So this matches nicely with what we would predict from our model. The other thing, there we go. The other thing that the model predicts is that it's actually very important, not just that you have this patch of myosin, but where it's localized in the embryo. So it's important for it to be offset from this position of maximum curvature. Because if you were to place it perfectly symmetric to this, maximally curved region of the embryo, you would actually both see a gradient in curvature in the normal kind of upwards direction, but also in the downwards direction. So you would expect forces that are pulling both up and down. And if you start perfectly symmetric to this position, those forces balance and you don't move at all. So experimentally, again, Drosophila is nice. You can basically have any mutant you want. We can use a mutant that has this original patch of myosin, which is normally kind of up on this top side of the embryo. And we can have, by again changing the dorsal and ventral patterning of the embryo, we could take this patch of myosin and really center it on the end of the embryo. And in some of the embryos, you get flow that looks relatively normal. Again, if we quantify the flow, we see that it's slowed down compared to wild type. And if we do, again, simulations where now we're changing the offset between the center of this patch of myosin and the region of maximum curvature, we can again predict that we slow down the flow and we should pretty much suppress the flow as we get to zero offset. But what's even more interesting is that this flow in the correct direction actually only happens in some of the embryos. There's also some embryos that we see on this bottom embryo that flow in the opposite direction, and some embryos that flow off into the third dimension, either into or out of the slide. So we believe this shows that just the slight difference in initial offset of the myosin is enough to direct the flow in different directions as it's responding to gradients of curvature in other directions. So with that, I'll summarize. So what I showed you today is that the motor protein myosin in embryos can contractile forces that are enough to generate symmetric tissue flows, but that alone can't drive polarized tissue flow. What you need is a difference in the apical and basal concentration of myosin, which creates a torque that leads polarized tissue flow of that patch of myosin away from regions of high curvature. And so the big takeaway is that in addition to just thinking about kind of genetic pre-patterning and where you have these localization of protein expression or protein localization, you also want to take into account the mechanics and the boundary conditions, the geometry of the system, which also impart critical information for embryogenesis. With that, I would like to thank very strongly my collaborators, Bandhan Chakraborty and Matthias Merkel, Thomas Lecrie, I did the work in his group. And as I mentioned at the beginning, I'm now starting my own lab at the Interdisciplinary Nanoscience Center. So with just the last couple of minutes I have, I'll give a very brief teaser of what I'm working on now, which is just to say that what I showed you in the previous slides was really thinking about kind of a cut through of the tissue, getting rid of all thoughts of the cells, really just considering a continuum model of the embryo. And what the model assumes is that the tissue is going to flow like a viscous fluid, which is very nice and works very well and fits the data. But we know that the embryo is actually composed of cells. The cells are highly adherent one to the other. It's an epithelial layer, so you can't break cell-cell contacts. So thinking about how the embryo is actually able to have these tissues that flow like a viscous fluid instead of stretching like an elastic solid is kind of what I'm focusing on now, and specifically really looking at the cell scale dynamics. If you look at what's happening at some of these cells as it's going through viscous flow, you can see that they're highly pulsatile, really dynamic. So understanding how these cell scale dynamics play a role in embryo scale flows, and specifically really trying to think of how the fact that these cells are consuming energy and that they're out of equilibrium, how that plays a role into the creation of these dynamics in the form that we see in morphogenesis. And with that, I'll leave you with my summary slide, and I'm happy to take any questions.